bud or budding process, it's done. Next one is actin cable. Okay, next one is actin cable. This is the yeast cell. This is called organelles. This is very, very important of yeast cell: mitochondria, nucleus, cytoplasm, cell membrane, and cell wall. These are very, very important of yeast cell structure, organelles. Okay. Next one, budding, it's done. Actin cable. The actin cables are bundles of 10 actin filaments that helps cells establish the polarized growth by providing tracks to transport secretory vesicles and organelles. Transport secretory vesicles and organelles. Organelles means mitochondria, next Golgi operators, vesicles, vacuoles. Okay, that is called organelles. Towards the growing part of the cell in yeasts. Next, secretory vesicles. What is the secretory vesicles? In the yeast, Saccharomyces cerevisiae, this is the best example of yeast cell. The well-documented classical pathway involves the translocation of proteins across membranous organelles. Okay, what is mean by translocation? What is mean by translocation of proteins? Translocation of proteins means translational pathway in budding in yeast. Okay, translational means post-transcriptional modification, that is called translocation of proteins across membranous organelles. Membranous organelles means already I told mitochondria, next Golgi operators, vacuoles. This is called membranous organelles, endoplasmic reticulum. Okay. Next, example, organelles means endoplasmic reticulum, Golgi complex, and vacuoles to the extracellular surface by means of secretory vesicles. This is called membranous organelles. Examples. Okay. Next one, yeast's nucleus. Next one, yeast's nucleus. DNA is found inside yeast's nucleus like in other eukaryotic species. Yeast also possess mitochondria, which are organelles that produce energy. Next, DNA means deoxyribonucleic acid. Yeast nucleus main function, gene expression and RNA processing, genomic replication and repair. This is the yeast nucleus main functions. Motor proteins means such as myosins, next to kinesins or dynein provide the energy necessary for motility. These proteins, motility, it's very, very important. The cell nucleus in the central organelle. The central organelle means nucleus. That organelle handles the duplication of the chromosomes during cell division and the expression of the majority of genetic information. These genetic information transfer into the bud. Bud means daughter cell. This is called nucleus. This nucleus genetic material transfer into the daughter cell. Okay, this is the main function of the nucleus. Nucleus main function. Bud, it's done. But it's done. Next one, actin cable also it's done. Secretory vesicles already I explained. Next one, fat globule or lipid globule. Okay, this is called lipid globule or fat globule. Look at here. Look at here. This is called lipid globules. Look at here. Fat globules or lipid globules you can observe in only microscope. Look at here. Lipid globules isolated from Baker's yeast cells contain mainly sterol esters, next phospholipids, as well as sterols, free fatty acids, and diglycerides as minor components. Triglycerol means three glycerol. Okay, tri means three. Next, fatty acids already you know. Uh, next, fat means cholesterol. Next, diglycerides. Di means two glycerides. As this is called minor components. Minor means minor components means small components. Okay. Next, this is the original peak of lipid globulins. Next, endoplasmic reticulum. Next one, endoplasmic reticulum. It consists of the nuclear envelope and the peripheral ER. ER means endoplasmic reticulum. ER, it is the short form, a single network of interconnected sheets and tubules, which in yeast is located close to the plasma membrane, where it is referred to as the cortical ER. Cortical ER means cortical endoplasmic reticulum. Look at here. Look at here. This is called endoplasmic reticulum. Endoplasmic reticulum main functions. ER performs a variety of functions, including protein translocation and folding, lipid synthesis, and calcium homeostasis. Okay, this is called endoplasmic reticulum main functions. Endoplasmic reticulum, it's done. Next one is Golgi apparatus or Golgi complex. Golgi operators, look at here. The Golgi exists as a system of dispersed cisternae. In the case of the budding yeast, Saccharomyces cerevisiae. Okay, the Golgi apparatus is known to modify and sort newly synthesized secretory proteins. What is the main functions of the Golgi operators? The three primary functions of the Golgi operators are the transport, next sorting and modification of both protein and lipid. And the protein composition of the organelle reflects these functions. These are three main functions of Golgi operators. Next, the Golgi operators is known to modify and sort newly synthesized secretory proteins. This is also another function of Golgi operators. Okay. Fundamental mysteries remain about the structure, operation, and dynamics of these organelle. These organelle means Golgi operators organelle. Important insights have emerged from studying the Golgi in yeasts. This is Golgi operators, functions and structure of Golgi operators. Next, mitochondria. Yeast mitochondria are indispensable for synthesis of such iron containing compounds as FeS clusters and heme. Fe means ferrous. Okay. As mitochondria As mitochondria are essential for yeast survival, they must be faithfully inherited. The proteins MDM10P, MDM12P, and MM1P form a complex required for mitochondria binding to the actin cytoskeleton and actin based control of mitochondrial movement, inheritance, and morphology. This is called mitochondria. Main function of mitochondria. The mitochondria's primary function is to produce energy through the process of oxidative phosphorylation. This is the main primary function of mitochondria. Okay, it's very, very important. Produce energy through the process of oxidative phosphorylation. Mitochondria provide energy to the cell by generating adenosine triphosphate. Adenosine triphosphate means ATP molecules through the process of OXPHOS. OXPHOS means oxidative phosphorylation. That is called OXPHOS in eukaryotes. Eukaryotes means in yeast cell. Yeast is the eukaryotes. Okay, don't confuse. Mitochondria provide energy to the cell by generating ATP through the process of OXPHOS. ATP means adenosine triphosphate through the process of OXPHOS means oxidative phosphorylation in yeast cell. This is the mitochondria function. Next, vacuole. Look at here, mitochondria also it's done. Next one, vacuole. Look at here. This is called vacuole. Yeast cell in another organelle, vacuole. Look at here. Vacuole. The vacuole of the yeast Saccharomyces cerevisiae plays an important role in pH and ion homeostasis and is used as a storage uh, compartment for ions. Okay. Next, vacuoles are the most acidic compartment of the cell and have a defined lipid and protein composition that supports role in protein degradation, ion and metabolite storage and detoxification. Vacuole have a three important roles that is very very important. First one is protein degradation, next metabolite storage and detoxification. These three it's very very important. Metabolites storage means antibiotics and pigments, so that is called metabolite storage. Next, detoxification means removing toxic substances. Okay, that is called detoxification. Detoxification means removing toxic substances. That is called detoxification. Okay, these three it's very very important. Vacuole main important role: protein degradation, metabolite storage, detoxification. Next one, cell wall. Yeast cell wall. The fibrous component of yeast cell wall usually consists of beta glucan and or chitin. Beta glucan or chitin both are same. Don't confuse. Next, N-glycosylated proteins form an amorphous cross-linking matrix as well as fibers on the outer surface of the walls. Next, yeast cell membrane. Look at here. The yeast biological membrane is composed primarily of phospholipids, next glycospingolipids, ergosterol, and proteins. These four components, it's very very important uh, of yeast cell membrane. It is the biological membrane is composed of cell membrane, phospholipids, glycospingolipids, ergosterol, and proteins. Next. Phospholipids are primarily structural components of the membrane and are essential to the viability of the cell wall. Phospholipids and the primary structural components both are very very important viability of the cell. Look at here, this is the plasma membrane. Look at here, this is the plasma membrane. This is called cell wall. It is made up of a manoprotein layer, next beta glucan chitin layer already atal beta glucan or chitin, next beta uh, glucan layer. Look at here, this is called manoprotein layer. This is fibrillar layer. Okay, this is called fibrillar layer. This is manoprotein layer. Next one, beta glucan layer. Next, beta glucan chitin layer. Next one, manoprotein layer. Last one, plasma membrane. Fibrillar layer on the outermost layer of the yeast cell walls. They are often glycoprotein fibrillars. Okay, fibrillar layer or glycoprotein layer, both are same. Don't confuse. Look at here, this is called manoprotein layer. Manoproteins were located in the outer layer of the yeast cell walls. Look at here, this is the fibrillar layer. This is called manoprotein layer and were composed of polymers covalently. Look at here, attach it to the protein backbone. They were one of the main component of yeast cell wall. This is called manoprotein layer. This is also called one manoprotein layer. Both layers is there. This is called one manoprotein layer. This is also called another manoprotein layer. Next, middle one, look at here. This is called beta glucan layer and beta glucan chitin layer. Beta glucan layer or beta glucan chitin layer both are same. Don't confuse. Beta glucan in the yeast cell wall is composed of beta 13 linked backbone and beta 16 branches. Look at here. This is called beta 13 linked backbones. Okay. Apart from beta glucan, the yeast cell wall contains mannan and chitin. Mannan means mannose and chitin. Already you know chitin. Okay. This is both layers is there. This is called beta glucan layer. This is called beta glucan chitin layer. Next, last one, plasma membrane. Look at here. This is called plasma membrane of yeast cells. This is called plasma membrane. Specifically, Saccharomyces species. This is the best example of yeast cell. Look at here. Forms the barrier between the cytoplasm and the cell wall. The plasma membrane consists principally of lipids and proteins. The plasma membrane made up of lipids and proteins in approximately equal properties. That is called plasma membrane. This is the plasma membrane or cell membrane. Both are same. Don't confuse. Look at here. This is called yeast cell. Yeast cell structure in different organelles is there. Look at here, this is called bud. Already I explained by budding process also. Next, this is called actin cable, secretory vesicles. Next one, fat globules. Next, Golgi apparatus. Golgi apparatus function also it's done. Next, this is called vacuole. This is the nucleus. This is called DNA, deoxyribonucleic acids. Next one, endoplasmic reticulum. Endoplasmic reticulum also it's done. Next, mitochondria. This is called cell wall. This is called cell membrane. Okay, this is called. These are called organelles. Organelles of yeast cell structure and organelles functions also it's done. Okay. Next one, yeast cells application. That is also very important. Next, I am going to applications of yeast.